cellular and molecular biology. I thank you all for joining us today. We are here today with Mansi Srivastava, a PhD student at CCMB who studies and works with stem cells. With her, we are joined by experts in stem cell biology from different research institutes of India. They're experts who study the biology of stem cells as well as use it for disease management. And with them, we will be discussing the scope and future of stem cells in India. Mansi will introduce you to the experts very soon. But before that, let me brief you on the structure of the discussion today. Mansi has a set of questions lined up for the experts. She will address those in the first 45 to 60 minutes. In the meantime, the audience can put up the questions in the question and answer box. Please do not put it in the chat box for our ease of picking the questions. Please keep your questions relevant to the line of discussion today. With that, let me give the mic to Mansi and start the discussion. Mansi, over to you.
Thank you, Somdeta. Good evening, everyone. So I am Mansi from Dr. Chandrasekhar's lab, fourth year PhD students, and it's good to have you all with us. So I'll just begin the session with thanking all the speakers for agreeing to join me. Today we have with us Dr. Dovijyoti from IGIB New Delhi. So he works with genome editing tools. We have Professor Sujata Mohanty with us from DBT Center of Excellence for Stem Cell Research, AIMS, New Delhi. We have Dr. Pavitra with us from CCMB, who works with neural stem cells and organoid development. We also have with us Dr. Subir Majumdar from NIAB, who is an expert in the field of animal biotechnology. Thank you everyone for agreeing to join me today with this discussion. So, in the process of my graduate studies, I have come to appreciate the remarkable potential of stem cells that they hold in both applied and basic research. There are a lot of recent research findings like organization of mouse embryo-like structures solely from stem cells. They are generated in a dish just based from the stem cell models. There are also studies which is reporting reversal of Parkinson disease phenotypes just by edited neuronal stem cells. Recently, there is a paper that says that pancreatic organoids have been generated which can produce insulin in a dish. So all these findings, they highlight the value of stem cells, that how powerful as a tool they are. Today, what we will discuss is how judiciously are we really using this tool. While the rest of the world is pacing fast in the direction of tissue engineering, organoid research, interspecies chimera generation, stem cell-based therapies, how good are we doing in these contexts? Where do we stand with respect to the technological advancements that the rest of the world is making in these fields? We will also discuss about the resources that we have in hand. So stem cell research was recognized as an area of thrust research by DBT in 2001, which is 2 decades back. Government of India also has a lot of encouraging programs for development of regenerative medicine. However, the same government regulations do not allow their applicability. We are stuck with the applicability of regenerative medicine in India. So if you, just for the audience, in India, stem cell therapy is only limited to hematopoietic stem cell transplantation, and there is no other regenerative medicine platform which has been explored. So we will discuss the need to rethink our policies and their regulations which we have. What are the ethical roadblocks that have to be overcome in order to practice regenerative medicine in India? And should we have a separate governing body for stem cell research in India altogether? Like we have a Department of Biotechnology. Should we also have a Department of Stem Cell Research in India? Also, another thing that I would like to highlight through this webinar is, as a developing nation, we always have a resource crunch. The availability of resources is always a limiting factor for the growth of research. However, how well can we partition our resources between fields of applied research and basic research? Should we prioritize questions pertaining to our healthcare needs? So with these aims and scopes, I would now like to ask what our panelists have to say about these issues at hand. My first question is from Dr. Subir. So, sir, my first question for you is, how well can we compare the growth rate of stem cell research in India and in abroad? Do you think there are differences? And if you think there are differences, what is the possible cause for them?
This is a question to me?
Yes, sir.
Okay. So I'll give you answer, but maybe other members can also add up if they have better knowledge. So stem cell research in India is, it began competitively well when specifically Reliance and Manipal institutes, which are the two big institutes, they parallelly started looking at the potential in terms of therapeutic potential. And actually, we competed with Americans in whatever repositories are made and cells are kind of filed with them. And there was very good interaction between Indian and American scientists at that time. And the vigor with which it started, it did not continue. A lot of it becomes very, very important, that the number of labs which started those research in USA particularly was much more higher than that what we started in India. I'm talking about 2000, somewhere around 2001 in my view. And then the stem cell research, people have started doing little bit of basic science, basic research, and institutes still kept on working. Manipal, Reliance, it is not that much now, but I think Dr. Sujata will be best to speak much about it because she is actively into clinical research. And I do not think it is lack of funding, because in DBT continuously there is a task force on stem cell. And Dr. Bala Subramaniam from L.V. Prasad Eye Institute had been its chairman, who was earlier director of CCMB. And a lot of grants were given. It is not that the money was not given. But I think one of the issues, major issues, were having proper guidelines for the therapeutic purposes. So that is kind of went into a lot of litigations and those kind of things. And so many people started doing it randomly in India. You know, there is no regulation. There was no regulation, and now nobody, kind of, not all clinics follow regulations. So people started believing us little less, because we will claim so many things out of the blue that you can, instead of doing knee replacement, you can do stem cell therapy for bone morphogenesis, and those kind of different kind of claim. From diabetes, somebody claimed. And also there are people who claim for diabetes related blood vessel diseases. And, you know, that is the reason the faith on that clinical science internationally got little reduced. But basic research is going on in various places. It is very nicely funded. Dr. Sujata is actually member of several such committees. She will be able to shed light on that. And we fall back in terms of competition. The reason being, I would personally feel the expertise required to do high-grade stem cell biology, really very high-grade stem cell biology, and also high level of international collaboration. I personally feel that that is lacking in our country. Everybody wanted to do their stem cell biology. Part of the thing goes in also that is stem cell biology, several things of it is patentable, because everybody who gets something very hopeful thinks that I will patent and become rich. And that's why so many people do not disclose also what they are exactly getting and how they are proceeding. But I think now it has spread to animal sciences also, and in large animals. I am talking about dogs and also buffaloes. People have started working on stem cells and cord blood stem cells and bone marrow stem cells. And in dogs, some of them in IVRI have done therapies also. So it's picking up slowly, but I am very sure part of the reason becomes secrecy for the lack of growth in Indian scenario and international collaborations, which is very much needed, because technology import is very, very important in stem cell biology. Otherwise, it becomes, even if you read a protocol and try to repeat it, not necessarily you are going to get the same endpoint as ideally one would get. So this is important. Frank collaborations at the international level. Funding is not an issue in India, and also people who do random stem cell biology and spread false claims. So foreigners also not little hesitant to come. That's my idea.
Thank you, sir. So what I gather is collaborations is where we are lacking the most, which is a very important point for you to raise. So I like to ask the same question from Dr. Pavitra also. So since you have recently joined as a faculty and you have like first-hand experience of how things were working outside and what challenges you're facing once you are beginning things here, would you like to share your experience about the pace of research?
Yeah, sure. So, just like what Dr. Majumdar said, it is basically we are concentrated in pockets. Okay, so we don't have, so most of the stem cell biology stuff that happens, about the big difference I see, for instance, between UK and India is that you have big consortia which work together, which I think we are in the beginning of it. Probably we are beginning to establish something like that. So you have Center for Excellence, for instance. I think INSTEM is one such center. But apart from that, it becomes really difficult if you're like an isolated island, because even within stem cell biologists, if you see, they are very territorial, like what he alluded to. So it's like, oh, I work only on mouse stem cells, I work on human stem cells, I work on hematopoietic, I work on neural. So it's not viewed as a community right now here. So that's a big drawback, I feel, which should be addressed. And if it is addressed, I think once we have good consortia across, I think most of these problems can be addressed. So, and also the vigor with which it started way back in 2001, when we didn't even start our PhD, for instance, at that point. So we knew that in 2009, 10, like a decade ago, there was a big boom and there were a lot of labs doing a lot of basic stem cell biology work. But then 10 years down the lane, if you see, it's very difficult if you just go online to even search what is the kind of repository we have from whom you can reliably source indigenous iPSCs, because we would like to work with Indian iPSCs, right? So that itself becomes difficult because we don't have a, or at least it's not available in the public domain to find out that who can give it and whatever. So it all goes through a known network. So that seems to be an issue. So if we can kind of, centralize all this, or probably put it together in the form of a consortia, then I think there is a scope that this will go forward much better.
So thank you, ma'am. So both the answers point towards two problems, which is we lack collaborations with the world outside, and within ourselves, we are very fragmented in the stem cell biology area. So that brings me to the next question, which I have from Professor Sujata. So, ma'am, I want to ask you, should India be focused only on application-based stem cell research, or we should be like dividing our resources between the basic and the applied research areas?
Thank you, Mansi, for asking this question. So again, in my view, all the great clinical advancements are with the whole handling with basic research, fine. So like, especially in the case of stem cell research, so we can't advance into the clinical application unless and until we have done basic fundamental exploration studies and basic research. So that what is Dr. Majumdar was saying earlier, that people jumped into clinical research without doing the basic research, right? So there are, I'll just give you an example. In case of adult stem cells, which can be used for translational application, there are multiple sources. You can have it from bone marrow or from the fat tissue or from the umbilical tissue. So you need to know how to isolate them, what are the basic intrinsic properties of them. They are good in diabetes, or they are good for cardiac work or neuronal work. We need to have a very strong fundamental research on them before we can go into the clinical application. So I think they should both go hand in hand. Definitely basic research will throw more light and will make their application much more precise, accurate, maybe faster. We might take 10 years to see the application of stem cells if we don't do basic research. But doing basic research, it can reach to the appropriate person. So I'll not say that stem cell is a magic bullet, use it in any diverse field of disorder or diseases. There are specific stem cells which are needed for particular disease or particular application. So I think both of them should go hand in hand. Basic emphasis should be on basic research.
Thank you, ma'am. So a follow-up question to what you said, what I have is the time that it takes from basic to translational research. that time is very stretched out. So what should we do that we can narrow that time down? So we have the basic research of the system ready, and then we can translate it, fast track the translational part of it. So how do we do that?
Again, a very, I think, pertinent question, Mansi. I'll give it to you in a two-way. Fine. If you see any technology in medical sciences, be it antibiotic productions, or a vaccine development, fine. Every technique has taken around 10 years, a decade to mature and come to application, fine. It is also applicable to stem cell research. So we cannot say that we can jump the guidelines or the path which it has to take before it can be go into clinical research. Fine. This is, I'm telling you, a generalized strategy. So how best we can utilize stem cells and regenerative medicine for the benefit of the human race? The answer comes from my previous speaker in like Dr. Majumdar and Pavitra. So that is like we need to have that networking. Fine. If we all can join hands together, somebody is expert in some area. If we all make a team, a consortium, then we can increase the speed of our work and also the scale. Fine. Then we won't be having those lacuna, which is required to clear those regulatory guidelines. You need preclinical research. You need the safety data. You need toxicology data before it can go into human beings. So I think joining hands together, making consortium, or sharing the expertise of other groups will definitely take us much further and much faster.
Thank you, ma'am. Okay. So my next question is from both Dr. Pavitra and Dr. Debajyoti. So how can regenerative medicine be best used in Indian context? So what are the challenges that we face in India with respect to regenerative medicine? Like, for example, a lot of research and funds goes into colon cancer research and generating intestinal stem cells on all these things. But that might not be a major problem in Indian context. We might have some other kind of diseases which are more prevalent in South Asian populations. So where do we stand with respect to that? How well can we streamline the regenerative medicine process in India?
Pavitra, go ahead.
So one thing which Mansi, which you correctly pointed out, is that a lot of what we have right now in India, at least with the stem cell regenerative medicine perspective, is blood related, hematological. That is what we are trying to do. But if you look at a lot of cancers, so even within India, like South India is more prone to this head and neck cancer, and the oral cancer is much more prevalent. So for these, then you start identifying which exactly is the target for you, and then you accordingly fund. Instead of just having it as one big umbrella regenerative medicine grant, regenerative medicine thing. So that can be something which can be addressed. But again, it all boils down to how good of an expertise you have, because someone who does, let's just say, a lung organoid, or someone who is involved in that, might necessarily not be an expert in understanding how you get down doing all the other types of organoids or stem cells. So that's something, if you have some kind of a good collaborative network, then you always kind of borrow your knowledge base, you expand your knowledge base, and you derive knowledge from them, and that's how you identify and go forward with it. So, I mean, that is my take on it.
Yeah, thanks, Mansi, for inviting. And I just add a couple of sentences to what Dr. Pavitra said, is that I feel that, as far as regenerative medicine is concerned, there are certain reasons why stem cells have not reached the clinic to the extent that we had hoped they would do, let's say 15 years back when the Nobel Prize was announced. And I think scientists are pretty much now aware of these issues, what are the problems, and that's why you see that, except for a very specific set of diseases in doable surroundings, if I might say, in the body, a number of clinical trials involving stem cells, pluripotent stem cells, is actually much low across the world. However, I think the advantages of the organoid technology is somewhere where the stem cells can be really put into use for regenerative medicine, particularly in Indian context, provided there is an ecosystem for the biotech industries to actually use them and do that in a collaboration with scientists. I don't think so. An organoid technology per se has to be reinvented in Indian settings. What it needs to be done is that with a focused and allowing of scientific groups and non-scientific groups, which are, let's say, the industrial partners here, if an ecosystem like that can be built, then drug discovery, toxicity, non-mouse experiments, all these can be really moved ahead. Because what we have the possibility of doing in India is we can do multiplexing. We can do a lot of things at the same time, which does not necessarily mean you have to spend a lot of time and effort to kind of work out the basic biology of some established organoids. So that is where, at least if you want to talk about therapy, this is something where it can actually have a lot of emphasis, preferably from the top, from the funding agencies. Unfortunately, the biotech industry is still very naive in understanding the full potential that stem cells and their technologies based out of stem cells have. So it has a lot of things to do, because one is that we have very few scientists who have been working specifically on this field, so the dialogue has been very little over the years. And secondly, we tend to we do not jump into something which is too ambitious, because we look for a very short term gain. At least if you're a company today, if I'm going to start a company, I will not start on organoids, because I don't know what I'm going to gain out of this in 3 years, right? So we need people who are able to take those chances, but most importantly, their chances should be secured by some kind of system which is very prevalent in the West, where you have startups coming up, then getting in the seed money for a couple of years, 3 years or so. And then if it doesn't work out, it doesn't work out. But you need to have that backing, both from the banking sector, from the government, and so on, that such ambitious plans can be undertaken. And if it is successful, then you have something new that can start in the country.
Thank you. So both of your answers point to two major things. One is the targeted funding, which we do not have in the Indian context. And the other thing is that the biotech industry is not as flourishing as it is outside. We are not really focused on it. So I would like to ask Dr. Subir for what you have to comment on these two points which have been raised by Dr. Debojyoti and Dr. Pavitra. So you're mute, I think.
Yeah, the biotech industry is not flourishing. The answer, I think, already Dr. Sujata has given, because unless we have a strong basic research with solid proof of principle and a bunch of committed people, determined and very well trained, committed and confident to display to the biotech industry that you come up and join hands with us, and we are going to definitely make something, because most of our research in basic science is towards publishing papers and not really collaborating with a clinician to do the hardcore clinical oriented work with a proof of principle, probably using animal models, which is guaranteed lacking in my view. There may be one or two such groups which can put hands in hands and work, but definitely you need to put your hands on clinical samples and use good animal models to work and show what you are doing is correct and what you are getting is guaranteed to work, then biotech industry will come, either in terms of therapeutic use, which is one arm of especially iPSCs nowadays, or you make organoids or you do cultures and then use that for treating the disease with screening various chemicals, biochemicals and factors, and probably advising that gentleman that you don't have to do the cell-based therapy. We found that if you take drug X, your problem can be resolved, because in vitro we have done your iPSC and we found a series of screen, one or two of them is wonderfully working. So he saves not only the cell-based therapy, but sometimes he also is prevented from the risk of cell-based therapy. That level of confidence, that level of innovation, where it's a very attractive innovation, unless that is shown to industry, industry will never jump, because this is a very new field and it carries risk of failure. That's a very big thing. And I would say best is to prove those things in animal models, because humans, you cannot directly go for experimentation, and there are now Drug and Cosmetic Act and rules, everything is mixed up. ICMR, Ministry of Health, I think Sujata will know much more. Maybe she has to cut through those axes to do what she wants to do. I'm not a clinician, but it's going on. And I know, for example, one of the cases, which industry will come? There was a case few years ago where somebody for, I think, cerebral palsy was undergoing some treatment, and these rules came in. And they said, no, stop the treatment. We'll consider this as a new drug. That means the drug controller of India will give permission. And the doctor said, this patient is halfway through treatment. And if I now wait for drug controller of India, that means I stop the treatment, this situation can worsen. Then the High Court of Delhi ordered, no, go ahead with your treatment. All those people who come and file a case, we will see case by case basis, and we can give permission to go ahead with treatment. In the meanwhile, let anything new to be controlled by the drug controller of India. You see, in such a kind of a confusing situation, no industry will come forward. See, if two people are fighting, you're not going to go and open your shop there to sell something. Because किसी के हाथ में चाकू है, किसी के हाथ में डंडा है, वहां आप दुकान नहीं खोल सकते हैं कि I want to sell something here. This is a wrong place to settle. That has happened to the industry. Biotech industry needs a lot of confidence. They have money, but before giving money, they need confidence. That confidence, I think, we have not yet built up, and that needs to be done. Or you need some risky biotechnologist with an industrial frame of mind who would really jump into this business and say, okay, I will cooperate with you. I will also be part of what you are doing and try to resolve your issues with my power, with my influence, because neither scientist, basic scientist, nor clinician is enough influenced as an industrialist is. So probably it should be a joint effort by all of them and less risk for the industrialist. That's one of the answers.
Okay. Thank you, sir. So in your answer, you've mentioned something about embryo models. So my next question is something along those lines itself. So there are a lot of hype these days about synthetic embryo models. A lot of people are developing synthetic embryo models in vitro for the kind of drug screen or small molecule screen that you're talking about. So how do you think where do we stand when it comes to synthetic embryo development research for all kind of species in India?
No, I still didn't understand. You are saying synthetic embryo model for drug screening, or synthetic embryo model for making a woman mother who is infertile or conserving species?
No, the first part. No, the first part.
Drug screening. So
yes.
I frankly speaking, I don't have knowledge of that. A synthetic embryo for drug screening, no, I do not have any knowledge of that.
Okay. Okay.
Maybe if Devjyoti or Sujata has that knowledge.
Yes, if any
synthetic embryo for drug screening. I mean, is she talking about generating pluripotent cells and then?
No, I think Mansi, your question is about blastoids, probably, right?
Yes,
something along
blastoids.
those lines. Yes. Something
yeah, blastoids are far away from clinic. It is more as a developmental model and studying just what are the maternal child relationships and how the embryo develops inside the womb. So it is at a very, very non-translatable level, and I think such kind of studies have been going on for some time. Recently, Jacob Hanna's group has of course done this breakthrough, growing them in these kind of rotating shakers and so on. Again, these are groups which have past of fantastic stem cell work. And I would say that falls under the ambit of the basic biology, not so much about translational, which I think is the scope of today's discussion, probably.
Yeah, I agree with that, Jyoti. So this is really a very nascent stage, and this is also trying to help in not the use of preserved embryos in during IVF. So this is also to take some time to use embryo beyond 14 days, which is mandatory earlier. So this are the technique to bypass frozen embryo and use maximum number of pluripotent stem cells for developmental biology right now. Yeah.
And also, I think it's still limited. I think there's an international moratorium for human beyond 14. So this will all be done at mouse level or
yes,
some
they
other
are
similar.
just thinking to increase the time beyond 14, like maybe I think up to 29 or 30 days.
Yeah.
So earlier it is you can use embryo
non-humans,
within 40. So this is just gaining momentum to increase the time and also the use of it.
Okay, thank you for the answers. The reason I was asking this question is a lot of this synthetic embryo research actually requires a lot of high throughput engineering systems. So that leads me to my next question is, where do we stand with respect to tissue generation or modeling 3D tissues when you have the tool, which is stem cells in hand? Are we utilizing it at the same extent as the people outside are utilizing? And where do we really lag in this kind of studies?
So I think for basically you're talking, I mean, probably you're alluding to stuff like organ on chip or, even making scaffolds and stuff. So we, at this point at least, I don't think there is an active communication channel open between some of the the engineering sciences and cell biologists per se. Yes, there is some limited cost. I'm not saying it's not at all there, but for instance, I think IIT Madras has it, IISc has it. I think you have few IITs who have this microfabrication facility, but it would be great if there can be a conversation, and then organ on chip would be a cheaper way to kind of do it instead of making. I mean, it's again a completely different ball game compared to a 3D organoid. That would actually be a complex 3D tissue for all the drug screening and everything, but for quick. I think I read somewhere that if you start making this microfabricated chips in India, you could get it down to 1,500 rupees. A chip would cost you 1,500 rupees, an organ on a chip. But if you import it, it's going to cost you around 150 pounds or something. So that's around 10,000 or 15,000 rupees. So I think if we start this active crosstalk, we should be able to do organ on chip, at least microfabricated devices and everything. But as such, using 3D bioprinting for vasculature, which people are routinely doing abroad, we don't do that a lot. I mean, I'm not aware of a lot of groups who are doing that yet. Unless you have people who are like-minded, who want to kind of go towards this discovery mode, right? That's going to be difficult.
Thank
Mansi, can I add a little bit what she has said? Like if you're talking about use of organ on chip, either to study the toxicology effect or any drug effect, or to study the biology of stem cells. There are few startup companies because Birex, which is a division of DBT, they fund these startups. So I know, because I'm in that committee, that few startup companies are there who are working on maybe, I think, liver organoids to check for hepatotoxicity. So they are using those high throughput system where they can have maybe drugs which are used for chemotherapy in cancer patients for their hepatotoxicity. So, but they are very few because they are not many. But I'm really glad to know that few startup young people are coming up to take up the work. Second question is regarding, I think, the scaffold engineering. Now, again, the momentum has gained. I think Sri Chitra Institute, they do a lot of scaffold and stem cell engineering. Then IIT Guwahati and AIMS and IIT Delhi have joined hands together to work on a few areas which we have got, like cornea in the eye, like a lot of corneal blindness is there, if we can make synthetic cornea. Second is trachea, because there are a lot of patients who have got either bullet injury or some injury to the trachea, they need trachea replacement. So we are making 3D trachea, and also the bones. So these are the few areas which we get feedback from the clinician, what is their demand or need. So I think groups of people are coming together, both scientists, clinicians, and engineers, to go into this. Is another like tissue engineering which you talk about. Yeah.
Thank you, ma'am. My next question is from Dr. Devajyoti. Sir, what are the ethical rules for genome editing stem cells for clinical purposes in India? So, can you suggest any iterations from the existing rules which can make it better and more relaxed?
I think the recent release of the stem cell guidelines and the genome editing guidelines pretty much covers most of the stuff. There's been, I mean, it will get better and better because it's still very nascent, right? They work on this, and also coming from abroad, what are the developments that are happening? So we are still kind of understanding what the genome editing does to cells at a very superficial level, I can tell you, and it is limited by our methods to understand these differences. For example, genomic rearrangements is a very understudied kind of a thing. So yeah, I think the rules are there. There are guidelines which are there. As far as stem cells are concerned, I think there is to be clear distinction made. Stem cell is a huge, it's a big term. It has a lot of things coming under it. Here we are not talking about iPSCs. We are not talking about pluripotent stem cells. I think there is an international moratorium on any editing for developing an embryo. What is allowed, of course, is things which are multipotent or unipotent, let's say, going to HSCs, maybe hematopoietic stem cells. Trials have already started, not in India, but abroad they have started, and also some of the others type of multipotent stem cells. And I think it's very exciting times to be in because a lot of people have started working on that, and we'll see how it goes. But as far as rules and regulations are concerned, I'm happy that both DBT, ICMR, and DCGI, CDSCO, everybody has now come together, understanding the importance of it and formulating something which is forward thinking. I would not have too much of questions or criticism there. I think Dr. Mohanty is also there in this committee. So yeah, I think it's so far it seems to be okay, but it has to be continuously reviewed and updated as per the developments that are happening in this field.
Just to add to what Devjyoti said, that this is a very positive and very encouraging sign that all the funding agency and the regulatory authority, they have come together. Otherwise, it used to be X funding agency or regulatory authority doesn't know what is the rule pertaining to. So CDSCO and DCGI has made a special committee to look into the gene editing. And I'm glad, I think we have seen one proposal editing hematopoietic stem cells for hemophilia. So that is also a very positive sign. So we are moving forward in that. So it's taking shape.
Thank you, ma'am. So this leads me to my last question, which is from all of you, actually. So can you comment on where stem cell research will be in a decade from now? And do you have any suggestions to add for the young investigators who are working in this field currently?
I'll speak first.
Yes,
because
sir,
I am least knowledgeable about this. So whatever
Most experienced.
I fail to say, Devjyoti and Sujata will fill up. So
no, you have the expertise. You have the expertise.
So I will say one thing. First, to continue what Devjyoti and Sujata was saying, that in addition to DCGI, also I think we have to consider RCGM of DBT, which actually regulates anything related to gene editing in the country. So we have a committee, and we have a committee for human sciences, animal sciences, and plant sciences for gene editing. And the rules are being framed. They were put on the websites for comments. That has also been received. And I think any day it can become final, because 2 times we have got the comments. And I think gene editing for the purpose of research, we have ensured that that is allowed, because we were, like I was heading the animal, and there were other people, I think Dr. Rakesh Tulier, someone heading the plant, and somebody heading the human. And we ensured that for the purpose of research, that should be always allowed in dish. It should not be restricted, but there are grades. That what are you trying to do? Are you going to insert a gene, full gene, or are you going to replace a gene, or are you going to just put a nick without inserting anything new. Based on that, there are rules and regulations. But everything is allowable under what we call as controlled trial, but not with human beings. So we should not forget RCGM's role, because RCGM is one who will talk about the danger of doing what you are doing. And there are a group of bunch of people who really, I think Rakesh was chairperson of still he may be chairperson of RCGM. But that RCGM custodial committee where gene editing rules are made. So one has to refer to that. And as long as looking in a decade, I'm very sure that we just discussed the loopholes of the system, that what are the caveats, that hands are not held together. And we have to catch hold of people who are 100% dedicated to stem cell research. So like I do so many things, one of those things will be stem cell research, because I never wanted to make that my 100% bread and butter because of the risk involved in that field. But what should be done, ideally, that if agencies like CSIR or DBT or ICMR brings 4 or 5 people who are retired but energetic, very practical guys, and make a committee and decide they will decide which people to be picked up and what should be given to them to work with one target. So people should not apply for grants. I think this committee of wise people without any personal interest in anyone, I will not name, but I have seen people in the committee going and funding their own students, own people profusely, increasing the fund by 5 crore rupees. I was part and parcel of that. I feel very sad. No, there should be sincere people, and they should decide which are the guys who are doing very good research, who has the potential, and the wise people should decide what research is to be done. And they will just hold these horses, lambe race ka ghoda jise kehte hain na, that horses which will win, and ask them to do a particular kind of research. They should pick the industry and say, come here, we are the mediators, we give you partial guarantee of this group of people. From beginning, you try to be with them, help them in finding out what kind of market demand is there for treatment of what. And you know that you will earn money if that treatment becomes available. Neurological disorders, I think, should be very easy because you have no other choice than treatment. If you begin with that, you have nothing to lose. And those kind of cardiac, I think there are so many alternatives of cardiac treatment. For a stem cell therapy, if a man dies, you have a trouble. But neurological disorders, you have nothing else. So if you can pick up the right first thing, which India wants to focus, and a wise group of 4 or 5 people without any personal interest, picking up the right kind of guys and make a stepwise target of 2 years, another 2 years, and another 2 years. I think in 6 years, we are there. From where in 6 years, I think this kind of treatment can be started on a clinical trial in a hospital. I'm very, very sure about this. Even earlier, we are all knowledgeable. We are very nicely placed in terms of technology, equipment, funding. I think only you need to have someone wiser and elder governing a bunch of very, very, very promising people and giving them separate. And that means some 2 or 3 basic researchers coming together, achieving one thing, maybe 3D organoids and maybe scaffold, whatever, and then 2 or 3 clinicians. And in my view, they should be all at the level of associate professors, not above. Not at all. Can be 1 or 2 assistant professors also. I agree, because they will work day and night to achieve that. Professors like us have partial interest. We are into 10 meetings and people like you call us. So we have to honor you also. So you need people who are little mature also, little responsible also. I'm giving you a perfect example of an associate professor. That should be level and age of person. That's also very, very important. They should work at the basal level, basic research. Parallelly, the clinicians of that age should also participate, who can work day and night towards not only attending to clinic, but finding out what kind of patients, patient histories, what kind of treatment is possible, if needed, some samples, with which basic researchers can do some basic research, and then some enthusiastic industrialist who is interested in that. I bet 6 years is good enough time to progress and reach up to clinical trial stage.
Thank you, sir. That's an excellent point that you make, that we should have a structured research model for us to grow forward in a very fast pace. I would like to ask Dr. Pavitra also the same question, ma'am. What suggestions do you have for the young investigators, and where do you see stem cell research in India in 10 years from now?
I completely agree with what Dr. Majumdar said. So I mean, I have nothing to add to what he said basically, because I was just listening to what he said. I think if people can come together and switch over to this discovery mode, like one thing which he kind of he kept on saying was that if we have too much of interest vested in too many areas, then that becomes really difficult. So if the focus of a particular lab is this particular technology development, or let's just say that understanding this particular stem cell biology or whatever, then if they can be brought together with a common, I mean, that's what I was talking about, having a consortium or having, focused this team grants or whatever. So if something like that can happen, then things will move forward. And 10 years is a good enough time for us to make progress, because we have a huge, rich genetic pool from which we are not competing with anybody basically, because the gene pool we have is completely unique to us. So it doesn't matter if other countries are making their own whatever iPSCs or doing their own screenings. We might end up coming with something completely new to, indigenous to our country. So that's something which we can always think about.
Thank you, ma'am. Dr. Devajyoti, what do you have to say further?
I have no advice to give to young people. I think I'm still learning, and I have nothing to add actually. So I think it has been covered more or less.
Okay, thank you. Thank you. Dr. Professor Sujata, would you like to add something to the...
I think everything has been said by Dr. Majumdar, and Pavitra has also added to that. Devjyoti didn't comment because he's still young, so he has a lot of potential. So, like, jokes apart, so what I will say, there are two parts. First is, as Dr. Majumdar said, we need to think and cherry pick few people who have potential and give them the task, fine, which has a schedule. Okay, he said 2 + 2 + 2. So that we are really concentrated and focused in our approach. We know what we have to do and what we have to deliver after that much time. Fine. Apart from that, I would also like to say, which is very important in terms of stem cells and regenerative medicine, is your interaction with clinicians. So this is what I have learned being here, so that we need to identify our own problems. Rather than reading papers from USA, Europe, or any other country that because they are doing it, I have to do it, we need to also have our specific need. Fine. Based on that, if we do the research, then there will be many takers of that technology, and there will be many beneficiaries also. Fine. I'll just give you an example. This is for my young people who are just venturing into stem cells and want to see the fruit of stem cell research. Towards translational. For example, in India, we have a lot of corneal blindness, fine, which is unique to India. Nobody else in the country will see the corneal blindness, because we have a lot of child labor who are working in dangerous factories. So they have a lot of injuries to the eye, maybe chemical burn or whatever it is. And they lost the vision. Otherwise, everything is okay. And we don't have many cornea donor in India. So can we do some use stem cell technology where we have started limbal stem cell transplant in the eye, and which is very successful and very efficacious. Second example I would like to give, vitiligo. That is the white patch on the skin, loss of melanocytes. Because we are so-called dark skin, so the white patch is a problem for us, not for maybe a Western population because they are fair. So likewise, we can see it's not a very debilitating disease, loss of melanocytes, but it reduces the confidence of the young people who have them in the visible area. We found in our skin OPD that we can restore melanocytes using stem cells. So I think interaction with the with the clinician will also guide stem cell researchers in which direction to move. Third will be don't stop to dream, like dream impossible. I would like to say that this is the area where you should not limit your dream. So don't think that we are in India, maybe third world country. I will not say third world country. So we are just developing country. I will also not believe in that. We have a lot of intellectual wealth. We have a lot of hardworking people. Think of impossible. So we can move into that also. Fine. So this will be my message to the young people.
Thank you, ma'am. That's a very important lesson for us to identify good questions and to have big dreams too and venture towards working towards them. Thank you, all of you. So this brings the session to an end. We have some questions from the audience. So this Azim is asking that USA has changed its stem cell regulations. How will that affect the research scenario in India?
I think that what specifically has been changed has to be mentioned here. Otherwise, it's a bit unclear.
Yes, okay.
Maybe about embryonic stem cells they are asking. I think earlier they were allowing embryonic stem cells to be used for the clinical purposes. Then they stopped it because of some adverse events which were happening in patient. Maybe I'm just thinking that he's referring to that. But again, few companies have been given FDA approval to carry forward the clinical research using safe embryonic stem cells, or maybe fetal stem cells if he's targeting to that. But I think India has learned from all of them, although we were a little late in starting our regulatory framework, but we have learned from them, and I think our guidelines are good enough, and they are becoming better and the best now, with a lot of brainstorming sessions. Yeah.
We also have one more question from the audience, which is to ask that do we know what will happen if there is genetic engineering done in animals or organisms? I think this refers to the livestock being engineered genetically. And what are the regulations for that? To what extent can we edit the animal species?
I think animal species gene editing, as I said, in the committee of RCGM, they have allowed it up to the laboratory conditions. So we are not putting any restrictions for this research. And it has to be done in a contained environment. That means, let us say, I am trying to do that work using 3 animals. And it should be really in a contained area. And almost everything you dream about is allowed in animals. Technically, animals cannot talk about their own demands, but there are ethical committees of institute and also CPCACA, which acts as a brain of animals' rights and ethics. So they cumulatively decide what kind of experimental things can be done with the animals. And RCGM DBT would allow that what kind of genetic manipulations you can do. There they have allowed almost everything doable, but in a limited trial basis, not to the field. Because before going to the field, you have to really study the whole genome of the animal after the manipulation and ensure that rest of the things are okay. Almost whole genome sequencing and everything, that nothing is hindered except for targeted gene editing, whatever you have done. Now, gene editing can be done in different ways in livestock. Let us assume that I myself have been working to generate very costly therapeutic proteins. I'll give a very good example for students. It appears that there are students also. That we are trying to generate very costly therapeutic proteins in the milk of animals. Because those therapeutic proteins are generated in fermenters, bioreactors, in very big industries of 4-5 acres, light, electricity, license, 3-shift duty, and several other maintenance costs. Obviously, the cost of the therapeutic proteins are very high, 13,000-14,000 rupees a vial for gamma interferon or something like that, which poor people cannot afford. But if we can make that, making livestock as a bioreactor. So I generate a transgenic cow, where under a udder gland proteins promoter, let us say beta-casein, we express human gamma interferon. So in the milk of that cow, gamma interferon will be generated, and you can purify that and make small vials and sell. So instead of 13,000 a vial, it may be produced in 150 rupees a vial, because my cow is industry, land, electricity, light, 3-shift duty, license, everything. So it's a live industry, and I'm spending almost peanut for it. So while doing it, we call it that we do genetics manipulation and we generate that transgenic animals. So we are working for it. But parallelly, we started thinking that the question of GMO will come, that if by chance this cow escapes and somebody gets hand onto it, it can reproduce the cow, number one. And number two, we do not know that because of the manipulation, after 10 years, we find that its progeny are more prone to FMD or brucellosis. And I don't know today. So we started thinking, rather than making a whole transgenic cow, where only udder gland specific promoter will drive a human gamma interferon in the milk, if we directly transfect the udder gland of the cow, then the animal is not transgenic. It will not transfer this change into next generation because it is not gonadal manipulation. So germ cells do not have this editing, but this cow will produce milk where I have transfected the udder gland, and the milk will have the human gamma interferon, giving us the same solution. Both are gene editing in livestock. The second one is much more less risky and acceptable because the standing cow is not GMO.
Yes.
So you see, in livestock, if you really put innovation also into it, you can get very quick practical gain. I don't know, industrialist will call in terms of industry, but I as a government employee and a scientist would call it a service to society, that very quickly a 13,000 vial will be sold at a rate of 150 to 100 rupees. So in animals, it is allowed, it is doable. But remember, if you do certain things in animals using transgenesis, let us say, for example, I produce some medicine or medicinal therapeutics in cow's milk, and then somebody objects that it has got, I am a Hindu, and from cow's milk, you are generating something which doesn't suit to my mentality. It is a product of cow. It is not milk. It is a human protein you made there, then I would say parallelly one should work, whether it is human or animals, political and religious sentiment should be parallelly taken into consideration. And it's the right time that people start having a constant discussion, as Dr. Bala Subramaniam used to always write about science articles in Hindu. Somebody should constantly write about the nitty-gritty of this, the rules regulation of this, the benefits versus weighing the caveats versus the benefits, and prime people to accept these things when they come handy for them in future. That's very important.
Thank you, sir, for raising excellent points. We have one last question from the audience, which is for you, Sujata ma'am. So the question is the stem cell research in India focused towards our healthcare needs, or whether it's not really focused and it's distorted?
So may I start, or Dr. Majumdar has to say?
You, ma'am, you have to start.
We are talking healthcare, you should start.
Sure. Thank you. So basically, initially what happened was, because there were a lot of news in the international literature about the use of stem cells, fine. So people started in a haphazard manner to do the work. Fine. At that time, there was no regulatory. I'm talking about early 2000 or 2003, 2 and 3, where a lot of report came the use of stem cells apart from the cancer area, which is well established. That time regulatory authority was also not synced with the clinical research. Fine, it was not regulated. But later on, I think AIMS was the first institution which started clinical trial, clinical research, rather than the clinical therapy which people use. Fine. So which was very well peer reviewed by all the ethics members. And then it was sent to a funding agency. Then we got all the money to do the clinical research where we didn't charge from the patient. It was because they were under the clinical research. And then it was a systematic one. We took 2-3 different types of cardiac diseases, maybe 2-3 types of neuronal diseases, as they said. We identified based on what is the need. We thought that if there are current therapies which are not enough to give relief to the patient, then there was a hypothesis that addition of stem cells might augment the current therapy. So that was the aim. It was not that you can totally abolish the current medical practices or medical.
That was the theory which we started working on. And then there were other institutions also which joined hands, and there was systematic research later on. But earlier part, there was none. So right now, all the funding agencies like DBT, ICMR, CSIR, RCGM and CDSCO, DCGI, they are all well aware of the guidelines, and which gives very little room for unorganized clinical research using stem cells.
Thank you, ma'am. So we don't have any more questions. So I would like to thank you all for agreeing to meet me online for my podcast. It has been a pleasure to interact with you all. And I will be stitching your responses together, and I'll generate a podcast which I'll share with all of you. Once again, thank you very much for agreeing and for being a part of this one new adventure that we are on to. Over to you, Somnata.
Yeah, and it was a very brave discussion, I would say. It was nice, bold, and candid. And we will get back to you once the podcast is ready. And thank you again on behalf of CCMB for being a part of this. Thank you so much.
Thank you, Mansi. Thank you, Somdat.
Thank you.
Thank you, Mansi. Thank you, Somdatta. And it is you have given us opportunity to meet all of us together.
After a long time.
We did a long time, yes. And then I met Pavitra also. Nice meeting you. And I know they are all my friends, Dr. Majumdar and Devjyoti. We know each other. Thank you so much, Mansi and Somdatta.
Thank you.
Thank you.
Thank you.
Take care. Thank you
very much. Take care.
Thank you.